welcome to the fifth of our 6 Cafe Sci talks this summer. My name is Beth Orcutt, and I'm a senior research scientist at Bigelow Laboratory. I'm filling in tonight for our regular host, Dr. Deborah Brock, President and CEO of Bigelow, who sends her warm regards to you all. Before I get started, there we go. A reminder that as a nonprofit research institute, the discoveries, solutions, and inspiration we create are made possible through the generous support of our sponsors. And I want to start by thanking each of our donors, our board members for supporting the work that we do. I especially want to thank HM Payson, who has been a wonderful partner for the lab over the years and has been sponsoring our Cafe Sci series. And HM Payson is a Maine-based financial advisor. You may have noticed closed captionings appearing on your screen as I've been speaking. If you don't see them and would like to turn them on, or if you do see them and would like to change the settings or disable them, please click on the CC live transcript button in your toolbar, and you can follow the prompts to change the settings. You can also use the Q&A button that looks like some little dialogue clouds to enter in questions, and we'll get to those as we have time. You'll also notice raise hand and a chat button, but we won't be using those features tonight. Now it is my pleasure to introduce Dr. Alex Michaud. Alex received his PhD in ecology and environmental sciences from Montana State University, where he studied life in ice covering Antarctica. From there, Alex moved to Aarhus University in Denmark, where he brought his polar microbe fascination to the Arctic. Alex joined us at Bigelow Laboratory in May 2019 as a postdoctoral scientist working with Dr. David Emerson. And this summer, Alex was promoted to a research scientist here at Bigelow. Alex is joining us live from Tulip Field Station in Alaska, and he will be telling us about the life of ice and the global influence of microbes in polar regions. Over to you, Alex.
Hi, everyone. Thank you very much, Beth, for that introduction. And thanks to you all for joining me today to learn about my research and plans for future work at Bigelow. As Beth mentioned, I am joining you today from Tulip Lake Field Station in far northern Alaska. Tulip Field Station sits about just north of the Brooks Range, about 10 hours drive north of Fairbanks. And since this is one of the first remote or distance Cafe Sci lectures, I apologize if there's any connection delays. The internet is usually really good up here, and so I hope it stays that way throughout this entire talk. Since I am at Tulip Field Station, I want to acknowledge that the Tulip Field Station sits on the ancestral hunting grounds of the Nunavut and occasional hunting grounds and routes of the Gwich'in, Koyukuk, and Inupiaq peoples. And we are grateful for their stewardship of this land. I'm actually sitting right now in Lab 1. You can't quite see it, but I've got a great view out of the lab window towards sort of the southern end of Tulip Lake. And the other thing that you notice in this photo is this vast extent of Arctic tundra. And what is invisible here is the permafrost that underlies all of this land. Tulip Field Station sits in an area of continuous permafrost, which means beneath an active layer that thaws seasonally down to about 20 to 24 inches, there is a layer of thick, hundreds of feet thick permafrost that does not thaw every year. Alas, I won't be talking about this permafrost all that much, but rather talking about glaciers and ice sheets and the microbes that live in amongst the glaciers. Now, glaciers and ice sheets may seem distant for many of you, wherever you're coming from in North America or the rest of the world. And even to me at Tulip Lake Field Station, sitting at 68 degrees north, as I look at the Brooks Range to the south, and I see these small glaciers, the remnants of past ice sheets, they still seem quite distant, but it hasn't always been that way in North America. About 21,000 years ago, the Laurentide ice sheet covered a vast majority of North America, including this red outline of the state of Maine and much of where I grew up in Minnesota, as well as where Tulip Field Station sits right now on the north slope of Alaska. And this ice sheet actually existed and maintained its some size until about 10,000 years ago, until it retreated all the way back into the high Canadian Arctic. And Maine's glacial history actually plays into the story that I'm going to tell you today about the life of ice. And as you can see, about 20,000 years ago, as I showed in the last slide, all of Maine was completely covered by the Laurentide ice sheet, meaning that all of Maine was below an ice sheet. And as this glacier retreated about 15,000 years ago, you can see the edge of this ice sheet runs almost along the coast of Maine. And there was this ice sheet ocean interface, which existed right about at coastal Maine about 15,000 years ago. And to look at that a little bit closer is this map that was produced by the Climate Change Institute at the University of Maine. And you can see we're highlighting the down east portion of the state of Maine. And to orient yourself a little bit more, here's Bar Harbor and Acadia National Park. And they put the University of Maine put this climate change or this ice sheet history map together with points along it that you can go visit that shows evidence of the Laurentide ice sheet in the past. And what I think is really cool is that about 15,000 years ago, this ice sheet edge more or less followed Highway 1 that I've outlined in red here. So you can actually drive up and down Highway 1 and visit some of these sites. Now, ice sheets and glaciers grow and shrink in the same way. Glacial ice is born up here when snow layers are come down, and through successive layers of that snow and through lots of time and pressure, turn into glacial ice and then flow out of the mountains through their tumultuous teenage years in this crevasse zone and flow out into the valley floor. And then the glacier ice dies down here where it melts or breaks off into chunks or simply erodes away. And actually, we can figure out whether or not a glacier is growing or shrinking by making a budget between how much is going into the top and how much is leaving the bottom, just like your checkbook. If you're putting more in, it's going to be growing. Now, I'm talking about glaciers here, and you see this very well-defined glacier. But an ice sheet is just another type of glacier. It acts in the same way. It's just much bigger and covers topography, so much so that it's big enough that it can cover full mountain ranges. So the same thing applies. Snow accumulates here, and through years and years of more snow accumulation and lots of pressure, it turns into ice. And all of that pressure forcing down, plus some heat from the earth, makes the bottom of an ice sheet actually quite wet. And there is a lot of water that flows beneath the ice sheet. And so in this cartoon, I've indicated one of the places that I'll take you to, a subglacial lake that sits beneath the West Antarctic ice sheet. I also like to point out this ice shelf here. And this is a unique feature of the Antarctic. And this is an ice shelf is where the ice sheet flows out and becomes thin enough and actually floats on top of the ocean. And this is an important point for the future of ice sheets. Glaciers and ice sheets are also now considered a unique biome, like the coniferous forest that we might visit in Maine. It has its own unique microorganisms that live there, like the coniferous forest has its unique plants and animals that live in it. And glaciers and ice sheets have their unique biome, mainly composed of microorganisms that live on the surface of the glacier, within the ice, and below the glacier, or what's called subglacial. And I'm going to be talking a lot about the subglacial microbes that I've worked with in the past today. Now, this habitat for microorganisms in ice is decreasing. In the Canadian Arctic, we see that glaciers are retreating along much of the Canadian Arctic. All of these dots indicate an individual glacier, and those dots that are in dark or warmer colors, so the reds, orange, and pink, are all retreating, meaning that they did that budget between how much is going in and how much is leaving, and they calculated that these are actually decreasing in size. One other way that I like to tell about how the Arctic is losing ice is from this study that looked at the number of skating days on a canal in Ottawa. And what they see is in the past 40 years is the number of days that you can skate on that canal has been decreasing. And to me, this one hits a little bit closer to home. Growing up skating on ponds in my backyard, and my father, who is hopefully joining the seminar tonight, in his 35-year career working at a suburb of St. Paul and managing parks there, they had lots of recreational ice rinks. And in his 35-year career, he also noticed that the number of days that they had to maintain those ice skating rinks was decreasing. And you can see that they projected this model forward towards the end of the century, and they had fewer skating days over the course of the century. Ice loss is also occurring in the Antarctic, but in a different way. And one of the more troubling ways that Antarctica is losing its ice is through its ice shelves. And remember, I told you before, the ice shelves are the parts of ice sheets that are actually floating on top of the ocean. They're no longer grounded on land. And this is important because these ice shelves are the buttresses to the Antarctic ice sheet. Just like the buttresses help hold up the walls of the Notre Dame Cathedral, these ice shelves help hold back the flow of ice from the interior of the Antarctic. And so as these ice shelves thin, they become weaker, and they cannot hold back the flow of ice as well as when they're thicker. Just like if these buttresses became much thinner, they would not hold up these walls of the cathedral as well. And so in my past, I've studied many and explored many icy habitats for microbes, and I'm going to tell you about two today. First off, subglacial lakes in Antarctica, and then the second part we'll head to the Arctic and look at fjords of Svalbard. And many times we don't actually look at Antarctica from the bottom. And so just to orient yourself, when you see the peninsula of Antarctica, that's headed up towards South America, Chile, and Argentina. And what we're going to be talking about a lot is the Ross Sea region right here. And if you were to head straight north from the Ross Sea region, you would come to New Zealand. And sometimes it's difficult for me to contextualize and describe the size and vastness of the Antarctic continent. It's until you actually fly over it, it's difficult to conceive of. But just to give you some idea with some numbers, the Antarctic and Greenland ice sheet together contain 99% of fresh water on Earth. And the Antarctic ice sheet alone, it takes up about 5.4 million square miles, which is equivalent to the United States and Mexico. Yet all of this area of the Antarctic ice sheet, and we still, until 2014, we did not know what lived beneath the ice sheet. And the area beneath the ice sheet is actually a very important comparison that we can make for ice-covered oceans for other places in our solar system. And one of those places is a moon of Jupiter, Europa. And Europa is very special because it has some features that would be good places reasons that it would be a good place to look for life. First, it has an ice covering to protect it from all of the things that would come in from space. It has a thick liquid ocean, and then it has this metallic core. And what's really cool about this metallic core is that as it spins around Europa, it actually flexes. And in that flexing, there's a lot of frictional heat that's produced. And that heat migrates out towards the outer parts of Europa. And so there may actually be heat and rock and sediments down here with an ocean above. And if we compare this photo here of the outer crust of Europa to Antarctica, it actually looks quite similar. So if we were to lift the ice off of Antarctica, what we would find is a lot of water. It's ponded in lakes. And actually right now there are over 400 described and documented lakes beneath the Antarctic ice sheet, with Lake Vostok being the largest and one that you may have heard of in the past. It's equivalent to a Great Lake in North America. And also the water flows in rivers. So the water collects and flows in these large rivers to the coastal ocean and ultimately the Southern Ocean. And again, we're going to focus on the Ross Sea region here and take you to two subglacial lakes in this back corner. And so if we zoom in on this southern portion of the Ross Sea region of Antarctica, we find two subglacial lakes, Subglacial Lake Mercer and Willens. And to date, these are the only two subglacial lakes that have been sampled. And I was a part of both of the expeditions that went to these lakes to collect water and sediment to study the importance of these lakes beneath the ice sheet. And our camp in 2013 for Willens Subglacial Lake looked like this. And what we used was a hot water drill. And what's special about this hot water drill is that it was drilling with sterile water. So we went through a lot of work to filter and UV irradiate it, and also pass it through heaters to effectively pasteurize the water before sending it down the borehole to drill through the ice. And we had to go through all of these steps to prove that our drill was very clean, because we did not want the samples that we were collecting from these pristine subglacial lakes to be contaminated with something from the surface. And so much of my PhD was spent studying how can we make the cleanest possible water, and proving that the system that we had come up with was producing clean water. And so it's quite difficult to conceptualize what it's like to drill through an ice sheet and go down a borehole to sample a lake. And so what I'm going to take you through is a video that was put together by some videographers that were with us on our Mercer Subglacial Lake project. And I'm going to kind of walk you down the borehole to Subglacial Lake Mercer. So what we first see is we're actually looking upwards. So we just actually went through the surface of the ice sheet, and we're looking back up at this very otherworldly pink colored UV lights. And everything that passed down the borehole had to go through this ring of UV lights to try to kill anything that might have made it past our cleaning protocols. So anything on the cables or instruments going down this borehole went through this UV collar to be cleaned. So we continue down the borehole. And eventually we reach the water. And now this isn't the water of the lake. This is where the water has actually risen up in the borehole. And now we enter the water-filled borehole. And if I were to continue this video for the entirety, we would be going about a half a mile down this borehole. And I think that would take up the rest of our time and actually be kind of a boring talk. And so I'm going to speed it up here, and eventually we're going to hit the base of the ice sheet. So this is basal ice, so it's indicating to us that we're getting close. And there it is. That is the base of the West Antarctic ice sheet, with the entirety of a half a mile ice going up this way and about a 30 feet deep lake below this interface. And now you're going to see something kind of running along the bottom of the ice that looks like little ants. Right there. Hope you saw it. Yeah, right there. And in fact, those aren't ants, but those are gas bubbles that are melting out from the base of the ice sheet. And this is important to remember for some of the microorganisms that I'm going to talk about in the next few slides. And so now, instead of looking sideways at this interface between the base of the ice sheet and the lake water, we're going to look down towards the sediments. So there we are looking down, and you actually see this little piece of hosing that we put on the outside to actually see how soft the sediments were when we looked at this video when we brought it back to the surface. So this is what the surface of the sediments looks like beneath a half a mile of ice. And so I want to thank the film crew that we had with us for some really excellent footage. And I also want to note here that these observations, while they were video, these are important validation for a lot of the glaciology methods that have all been done from the surface, but many times not actually validated by going into the subglacial environment to prove that there is water beneath the ice sheet. And so this video alone was instrumental in helping out validate some of the assumptions that glaciologists use in their modeling. And we're going to go to the next slide. There we go. So to talk a little bit more about the life that we found in these subglacial lakes that are permanently dark habitats, one of the big questions that we had is, what is the base of this food web? So these lakes are cut off from any light, so you can't have plants helping this ecosystem along like they do on the surface. And one thing that we found was that subglacial water contains oxygen. And this was a bit counter to what a lot of people thought before we actually entered a subglacial lake and sampled it. And this subglacial water contains oxygen because glacial ice has little bubbles that are filled with ancient atmosphere. And these little bubbles are how scientists have pieced together past climate history by studying the atmosphere that's trapped in these bubbles. So since there's old atmosphere in these bubbles, that means there's oxygen, nitrogen, and CO2 that's been preserved. And when this melts, like you saw in that video, those gases are released into the water column. And why is that good for microorganisms? Well, one in particular that I would like to highlight are the iron oxidizing bacteria. And so they can use the power of iron that's found in this very common mineral called pyrite, or many of us may know it as fool's gold. And so there's a lot of energy stored in fool's gold when a microbe can use it when oxygen is present. And these microbes can actually use CO2 and the power of iron and oxygen together to make their own cellular material, so much so that they can grow and divide and also produce excess organic carbon, which forms food for other microbes. And so this is why we would call them the base of the food web, because they're using energy from minerals to produce food for other microbes and themselves. And the other byproduct that they produce from their life is rust, iron 3. And this comes into play a little bit later. So remember this, that iron oxidizing bacteria make rust as part of their life. And this is how that iron or that rust looks like. If you've attended previous Cafe Sci lectures, I'm sure you've heard Dave Emerson talk about the iron oxidizing microbes that he studies. And this was one of the big draws for me to come to Bigelow, was to work with Dave and help get some of these very important subglacial iron oxidizing bacteria into culture and to study them. They make these beautiful structures from these sheaths that are all ultimately made of rust, and these helical structures as well. And what I've put in here is a picture of what they look like when we actually grow them in the laboratory. They form these beautiful bands of rust in these tubes. And so one of the main goals that I would like to do is try to get some of these subglacial microbes that I just talked about into culture and work with them in the laboratory to really study what genes they have and how they live and how they make all of that organic matter for the rest of the food web. But we haven't just found those iron oxidizing microbes beneath the Antarctic ice sheet. In many of the glaciers from around the world, all of these green and blue circles that you see, we've also found iron oxidizing bacteria. And so they're actually found all over the world beneath glaciers. And not only that, but they may have inhabited Maine during past times when the ice sheet extended all the way down to the coastline of Maine. And we can see evidence for Maine being a subglacial environment by these scratches or grooves on bedrock in coastal Maine. And so if you've ever been walking along the intertidal area and seen these long grooves in the bedrock, that's evidence of a subglacial environment. And so when I see these, I like to stand on these rocks and think about having a mile or so of ice above you and what microbes may have lived here 15,000 years ago. And what's even cooler is that just down here in the coastal mud, we find a related iron oxidizing microbe that I'm sure you've heard Dave talk about in past Cafe Sci lectures. And with that, I would like to pause and see if anyone has any questions from part one about subglacial lakes in Antarctica.
Thank you, Alex. This is a reminder to our audience that if you have a question, please click on the Q&A button at the bottom of your toolbar and you can type in your question. Alex, a question from the audience. How long does it take for the hole that you drilled at Subglacial Lake Willens to close up after you drilled it? And can the same borehole be used to sample over long periods of time?
Yeah, that's a good question. It's actually coldest and freezes the fastest at that. Remember when we first went into the water, that was about 150 feet below the surface. Just like if you've drilled into an ice when you're a lake when you're ice fishing, that water kind of rebounds up in the hole. And that layer right there that always freezes over fastest when you're ice fishing is where it really refreezes the fastest. And so the borehole will stay open for about 48 hours, and then it really starts to freeze back. You have to remember the middle of the ice sheet, about halfway down, is about minus 25 degrees Celsius. So it's really cold. And so that water will freeze back quite quickly. So after 48 hours of running our sampling equipment up and down, we have to pause, put the drill back in and go down and actually ream that hole so that our tools don't get stuck in the hole.
Sounds tricky, and no fish. Alex, another question for you. Can you tell yet if the microbes that you find in the Arctic and the Antarctic are related?
Yeah, they are actually. They are from the same genus, which means so they are very closely related. They're like two different species of maple trees. They're kind of in that same. All maple trees are in the same genus, and so they're that level of related.
Great. Got a couple pyrite related questions for you, Alex.
Yeah.
One is the pyrite that you mentioned finding in the subglacial lake. Is that on the surface of the lake floor or is it in the water?
That would be in the sediments. Yep. Glaciers are really, really good at grinding and crushing anything underneath them. And so what's really good for the microbes is the glaciers grind that pyrite really, really finely. And when it's ground really, really finely, there's a lot of surface area for the microbes to attach to and use that energy. So it's a good place to look for small pieces of pyrite. I had to look for them underneath an electron microscope to find them.
All right, well, this is a great lead into the next question is, does the pyrite get used up over the course of millions of years?
That is a great question. I did actually do some extractions of pyrite from Mercer Subglacial Lake, and it does appear that there is that it is being used up, but I won't go so far to say that it's completely used up, but I think I can see a decrease in the amount as we go down in the core. So I think there is a decrease in the amount with time.
Okay. Switching gears to think about the gases that you're finding in these subglacial environments, there's a question about whether there's any methane in those bubbles.
Yeah, well, as we know, ancient atmospheres did have some amount of methane in them, but actually most of the methane is made from microorganisms much deeper in the sediments. And so that methane is actually diffusing up from deep sediments below the lake. And it's actually a very good energy source for microorganisms at the sediment water interface. So yes, there is methane in the lake water, but it's very low because most of the microbes in the sediment have actually eaten it.
Okay. Well, on the other end of the spectrum and thinking about oxygen, how much oxygen do you find in those waters, and is it saturated?
Yep. So they're big differences. Subglacial Lake Willens was much smaller and shallower. The analogy that I would use for Willens Subglacial Lake is, so this one right here, I would say that one is more like a sort of a wetland pond, maybe in the Mississippi River Delta. And this one had low oxygen, so about 15% of what can be dissolved in water at the surface. So yeah, and then Subglacial Lake Mercer here was a bit deeper, and so that was more of an inland lake, and that had very high oxygen. It was actually super saturated with respect to oxygen. So very different oxygen environments in these two different lakes. And we think part of that is due to the fact that there's a lot more flow from different lakes upstream into Mercer Subglacial Lake compared to Willens that doesn't have any lakes upstream. It just has a small drainage channel, then the lake itself, and then that lake drains down to the ice-covered ocean.
Okay. Alex, a question from the audience. What energy source is used for the drill?
Yes, it is a diesel-powered generator that runs essentially the water heaters that they use at car washes. That's what heats the water, and a lot of it is all diesel-powered.
Right. A question about the iron oxidizing bacteria. Is there anything that feeds on the products of those iron producing bacteria?
Yeah, one of the more significant findings that we made was that those, we weren't able to track down microorganisms in the lake, so in Subglacial Lake Willens, that actually ate that carbon. But what we found is that when the water flowed down to the ocean, the ice-covered ocean, the microbes here were very much starved for carbon. And so the organic carbon, the excess organic carbon that flowed downstream and into this dark ice-covered ocean here, was actually like a fertilizer. They needed that carbon in order to start growing again. So it actually fed something downstream, more so than we think in the lake itself.
Okay. Alex, one final question for you, and thank you so much to our audience for submitting such great questions.
Yeah, thank you.
And we'll have another Q&A at the end. Have you found any multicellular life in these subglacial lakes?
Yeah, that's a common question that we get, and we have not found good evidence yet for multicellular. All the life so far has been bacteria or archaea, so single-celled organisms. There are fossil evidence of diatoms when the West Antarctic ice sheet was much smaller, but those are fossil evidence, and so those are no longer living. But we looked for DNA evidence of non-bacteria or archaea, so multicellular organisms, and we did not find any. That doesn't mean that they're not there, but we're still looking. But as of now, we have not found any.
Great. Well, thanks for fielding those questions, Alex. Go ahead and take over the next part of your talk.
Yes. Excellent. Yeah, thank you very much for those great questions. I look forward to another good session at the end. But we're going to transition now to the north and the Arctic. And so just to orient yourself again, here's Maine. This is a bit of an oblique view at the Arctic. And again, I'm up here in northern Alaska, and we're going here to Svalbard. And mostly on the west coast of Svalbard. And the fjords that I've studied, I've outlined here in red. And I'm mainly going to be talking about this fjord here. It's called Kongsfjorden. These are all Norwegian names because the archipelago of Svalbard is a Norwegian claimed territory. And the first thing that I want to mention, as I did in the introduction slides, that the high Arctic habitats are changing, meaning we're losing glaciers to retreat. And this is happening in Svalbard on the west coast. And I happened to capture a photo as we were on approach to the research station that corresponded quite well to an aerial photo that the Norwegian Polar Institute took in 1936. And so this ridge here is this same ridge right here. And you can see that this has retreated probably 3 to 4 miles in these 70 years. And in fact, when we sampled right here, it would have been about right here in 1936. So 70 years ago, this sample site would have been subglacial. So the point here is that these fjords are losing their glaciers due to retreat. But what am I actually studying in the high Arctic fjords is their sediments, and in particular, the carbon cycle of the sediments. And this is important to study because the sediments are long-term reservoirs of carbon. So carbon comes in from the land, from glaciers. It's also produced in the surface waters of oceans and lakes, and then settles out onto the sediments. And whether or not carbon is either stored for millions of years in the sediment or returned to the atmosphere is dependent on how much carbon is eaten by microbes, which then produces CO2 or methane and returned back to the atmosphere. And so understanding this balance of how much carbon is eaten in the sediments is important to understanding the carbon cycle. And not only that, but fjords are actually quite abundant, especially in the Arctic. And there are some quite close to Maine as well. Again, here's Svalbard. And Arctic fjords are not only abundant, but they're an unknown in the global carbon cycle because we haven't measured this as intensively as we have the coastal ocean or other parts of the ocean. And so the carbon burial is in these fjord sediments is actually changing as glaciers retreat. And so how does carbon burial change as glaciers retreat? Well, fjords are unique because they're under very strong influence of two very different inputs. The marine waters further out in the fjord near the mouth, so near the open ocean, have a lot of phytoplankton and light so that those phytoplankton can reproduce and make biomass and make these big blooms. And that phytoplankton then settles out to the sediments. About midway through the fjord here, you have a mixture of glacial material and phytoplankton. And this is not a great place for phytoplankton in the water column because these glacial plumes can be quite turbid, meaning that there isn't a lot of light that gets through them, but they also do have some nutrients. So some phytoplankton do live here and take advantage of the nutrients in this glacial plume. And again, they grow here, but when they die, they settle out to the seafloor. And in the middle of the fjord, the sediments are composed of kind of a mixture between the phytoplankton and the glacial material. However, when you're really close to the glacier, all you have is glacial material falling out to the seafloor. The amount of phytoplankton here is very limited because, as you can see here, this bucket, you can barely see it in the glacial plume. So it's very difficult to photosynthesize when you can't get any light through the water. And so these are very dynamic habitats because they integrate these two different sources, the phytoplankton and marine from this side, and all of the glacial inputs from the glacial side. But again, I'm studying the sediments. So what does that actually look like? So again, here's the marine side, and here's the glacial side. And these are sediment cores from two representative fjords on the west coast of Svalbard. And you can't quite see a difference in this transect of cores, but when you actually look at the microorganisms and the chemistry in them, you start to see the differences in carbon burial. And to figure out how much carbon is buried in sediments, we need to measure how fast these microorganisms are eating the carbon that was deposited from phytoplankton and other sources. So what we do is we go out and collect sediment cores. This is my colleague from Denmark, Hans, and also Katja Laufer, one of my other postdoc colleagues. And we spent quite a bit of time in the fjords together, pulling up mud from the bottom. And we did these incubations to measure how fast the carbon is being eaten in the sediments and turned into CO2 or methane. And so to measure the influence of the glacier and marine influences on this sediment, we collected a transect of samples, which are indicated by these yellow dots. And just like the Bigelow magazine is named Transect, this is sort of what it looks like when you would do it in a fjord. And again, the marine influences are coming from this way, and the glacial influences are coming from this way. And so what we actually found is where these two influences come together in the middle of the fjord is where microbes are eating the carbon the fastest and where you have the least amount of carbon burial. And we like to call this the Goldilocks zone. I think in the paper we called it the sweet spot, but I also think Goldilocks was an appropriate alternate name. And we think that this is a Goldilocks zone because the material from the glacier and the marine influence come together and they fall to the seafloor at just the right speed where the microbes can eat it, not only the fastest, but the most optimally based on how fast it's falling to the seafloor. So I would say that was our major conclusion from this work. But some of the future questions is, one of the big things that's going to happen in the future is that these large glaciers that end in the marine environment will ultimately retreat so far that they're back on land. And so is there more or less carbon burial when glaciers retreat onto land? And Svalbard is a great place to study this. because there are these paired fjords where glaciers end on land, and we can study the sediments in front of these land terminating glaciers right next to sediments that are under the influence of these marine terminating glaciers. One thing we can also do is compare it to places like Maine that have been deglaciated for a long time. And these marine sediments near retreating glaciers can be found in Maine as well. So by this star near Augusta is where marine sediments were deposited near or in marine water, but near the edge of the ice sheet. And so you can see this in this blue-gray clay, and then also this lighter brown clay. These are all marine sediments that were deposited in close proximity to the end of the edge of the ice sheet. And so this is another neat connection to the environments that I have studied in the Arctic and the glacial retreat history of Maine. And so to summarize the sort of big points of the work that I've done so far is that iron oxidizing bacteria are really the foundations of the food web beneath the Antarctic ice sheet. And continual monitoring of microbial activity in fjord sediments is important for understanding and measuring the complete global carbon cycle. We don't want to neglect all of that marine sediment that's in Arctic fjords. And Maine once hosted these glacial ecosystems like the polar regions do today. And so next time you're out exploring coastal Maine, I hope you pause and think about the microorganisms that may have been inhabiting Maine when it was covered by an ice sheet. But I forgot to tell you why exactly I'm in the Arctic. And like many other things, it's because of the iron oxidizing microbes. And the Arctic, and especially the tundra of northern Alaska, has a lot of them. And remember, I told you earlier that microbes make this rust as part of their life. And you can see it here in the microscope image. But when you put a lot of those organisms together, what you get are these biofilm-like structures here that actually come together to form a very nice reddish, rusty mat. And so I like to say that the tundra is rusting all over. And when it's not in this surface water, if you kind of squish your feet in the real soggy plant material, you get this very blood red water that comes up. And that's all due to the activity of iron oxidizing microbes. And so at Tulip Field Station, we have a very nice platform for doing our research. Right out back of camp, we have all of these boardwalks that extend into the tundra. And there is so much iron scattered throughout this landscape that I don't need to drive or fly anywhere. I can just walk out the back of Lab 1 that I'm sitting in and get into the tundra and have plenty of iron to work with and many iron oxidizing bacteria. These boardwalks are important to the tundra. I once heard the tundra described as when you walk across the tundra, it's like you randomly rolled out bowling balls across your yard and then threw the shaggiest 1980s shag carpeting over top of it and then saturated it in water. And that's what it's like to walk over, because there's so much water and condensed plants that it's very difficult to walk over. We affectionately call it tundra slogging. But more about what I'm actually doing up here is that these iron mats that form are really sticky to phosphate. Phosphate is an important plant nutrient. All of these plants need phosphate, and there's just not enough of it in the tundra. And so these iron oxides here, this rust, competes for phosphorus with the plants. And so understanding the iron cycle and how this rust is formed and broken down will help us to understand how the phosphate moves across this tundra landscape. And one way we're doing that is both directly sampling the iron oxide mats, but also using drone imagery to to actually map where the iron oxides are. And in this example drone imagery, so this this bridge over the river here is this bridge here. And so if you look at a drone image, you can actually see all of the rusty mats in this standing water here. And these are sites that we've studied just out back from Tulip Field Station. And it's even just below the surface of the plant layer too, in this nice rusty red band. So even where you can't see it in the surface water, it's also hidden below the surface. So iron is everywhere, and the iron oxidizing bacteria are everywhere in the Alaskan tundra. So if I were to repeat my take-home messages, the one that I would add is that iron oxidizing bacteria are ever-present feature in these icy environments. And with that, I have a lot of thank yous working both in the Arctic and the Antarctic. I will, thanks to everyone, all of my colleagues in Denmark and the field stations that I've been to, and both of the large field teams that I've been a part of, as well as all of my funding. And with that, I will take any other questions that you have.
Excellent. Thank you so much, Alex, for showing us a real-time data collection from Tulip. We have a couple of questions coming in from the audience. So just a reminder, you can use the Q&A button to type in a question and I'll read it aloud to Alex. The first one goes back to the data you showed us from Kongsfjord in Svalbard. And you had showed two sets of cores, some photographs of cores. And the sets looked kind of different, like maybe they were more or less oxidized. Can you talk about what those differences are?
Yeah, what we were, this is from Kongsfjord, and this set of cores is from Krossfjorden, and they are two, so this is Kongsfjord here, so this was the top red cores, and this is Krossfjorden here, and those were the gray cores. And most of that difference is due to the bedrock that the glaciers are crushing. There is some very iron-rich sandstone that the Kongsfjorden glaciers crush, and so that turns the sediment this pinkish red color. Whereas there are much different rocks that the glaciers crush and deliver material into Krossfjorden. So that's the main difference. We actually see the same patterns in both of these sediments, and it was important to us to study those patterns and to show that no matter what the geology was doing upstream of the fjord, we found the same patterns. So no matter the differences in geology, the patterns in that Goldilocks zone still occurs in these very different sediments.
Great, a transect of transects.
Yes.
Another question for you now, thinking about the tundra setting in the Alaska and maybe even northern Canada. How rapidly is the tundra permafrost melting and making it difficult to maintain infrastructure, including oil pipelines, I guess, particularly in Alaska?
Yeah. The pipeline, the first comment I'll make is the pipeline and the road, that the Dalton Highway that helps to service the pipeline are ever-present features anywhere you go in near Tulip Field Station and anywhere you can get to. It's sort of the ever-present landmark. And there was actually, I heard some stories about places that the pipeline is actually starting to shift a little bit due to permafrost thaw. But there was actually quite a bit of ingenious engineering that went into it to make sure that it would stand the test of time. But I think what's more concerning is that a lot of the native communities and the indigenous communities of coastal Maine are having to relocate because of the thawing permafrost. And that's on the coastline where wave action really accelerates the rate of permafrost degradation, because that water is splashing onto the permafrost and helping to melt it faster. In this area of Alaska, what's happening is more of a sort of a continual deepening of the active layer. And so what I mean by that is every year, every year this active layer gets a little bit deeper, which means the permafrost is slowly melting. And so that's one of the main ways that the permafrost is melting around here. I think I got at that question. Did I miss some part of it?
Yeah, I think that covers it. Thanks.
Okay.
Thinking again about glacier settings, moving away from the tundra, are there any areas where glaciers are expanding or growing?
Yeah. Let me pull up the figure from Antarctica. You'll see that the ice shelves on this side of Antarctica are actually in blue, and that means the ice shelves are increasing in thickness. And that mainly has to do with ocean currents, in that the ocean currents are still cold over here, whereas over here the ocean currents are quite warm. And this is a bit concerning because West Antarctica is prone to collapse, and with all of these ice shelves thinning, that is a real threat. There were a few in the Canadian Arctic that were growing, but it really, I'd say globally, it's a very low percentage, 1 to 2%. The majority of them are decreasing in size. And if they're not decreasing in overall length, they're thinning because that budget is going in the wrong direction.
Okay. Alex, a question for you, going back to Kongsfjord again, and the Svalbard work. A question about the Goldilocks zone that you mentioned in the middle of the transect. What are the specific things about that environment that make it favorable for the microbes to eat carbon, and is there more or less carbon burial there?
Yeah, so here, what happens is the glacier, so a lot of this carbon is broken down without oxygen. So microbes that breathe sulfate, iron, manganese, all of those sorts of what we call terminal electron acceptors. So they can eat carbon and breathe elements that are not oxygen. And the glacier influence that settles out here has a lot of iron and manganese in it. And so you can actually, in this area, you can run more types of microbes than you can over here. And so it's sort of like the buffet is sort of the same in this region, but you just have more types of microbes coming to the buffet in this region, and they're all eating at the same time. And so what that means is that you get less carbon burial because more of it is turned into CO2 and then can go back into the water column and ultimately to the atmosphere.
Great. Alex, I think this will be our final question, although you're getting some comments here that folks are excited to go looking for some glacial features around Maine and the
Wonderful.
houses. Our final question for you, you mentioned that iron oxidizing bacteria are the foundation of the food web in the subglacial lakes in Antarctica. What is the impact of iron oxidizing bacteria on the food web in the Arctic tundra? Do you know yet? Yeah,
so if I go back to this idea of this rust that the iron oxidizers make here, their role in binding phosphorus to the rust that they make impacts how much phosphate the plants get. And so I don't know if I would call them the foundation of the food web up here, but they're more maybe a nuisance to the plants because they can steal the phosphate away from them. So they can modify those nutrient cycles in this landscape. So understanding their distribution and how much phosphate is bound to these iron oxides is important to understand in a changing Arctic.
Great. Well, thank you so much, Alex. I'll give you a little clap for the audience who you can't hear. It was really great to hear about your work today. Thank you for sharing it with us.
Thanks, Beth, for moderating the questions.
Yeah, great. And for those of you still online, I hope you can join us next week for our final Cafe Sci of this summer series, where we'll be hearing from Dr. Rachel Sipler, who is our newest senior research scientist at Bigelow, and she started in July. She's going to be sharing with us her aims to promote healthy water conditions in the Gulf of Maine, while working with, learning from, and providing insights to the fishermen, farmers, resource managers, and residents who need them most. We thank you all for joining us tonight and for caring to learn about our oceans and what we're trying to do to understand and protect them. And we hope to see you next week. Thank you so much.